I'd like to thank the KL University President Satyanarao Garu and our Jagannath Rao Garu sir for having me here. Today I'm going to talk about computational pathology at eHRC. eHRC is our Electronic Health Research Center at IIITB. I'm with the data science group at IIITB. We are proud to have a collaboration with NIMHANS, where we work with the Department of Neuropathology, and we have a short team now. I'm going to talk about the work that we are trying to do here in neuropathology. So these are the topics I'm trying to cover. We'll talk about the background. We'll also talk about the workflow for computational histopathology, and we'll talk about what we call whole slide images, and we'll talk about how to do tile generation and how to curate a data set, and we'll also talk about the AI models that we use, and we'll talk the future work and the emerging trends in computational pathology research. If you see, all that we have seen now is a lot of radionomics work. Fundamentally, they are images and they augur well for processing by image processing and computational methods. Here we are talking about computational pathology here. It is an interdisciplinary science that augments development of computational approaches to analyze and model medical histopathological images. Histopathology, as we know, is having biopsy slides and watching under a microscope. Now, if that can be digitized, it brings a lot of data for analysis, and we can also do multimodal development. So there are typically there are these tasks that can happen in computational pathology. One is the detection, typically binary detection, malignancy, yes or no, and other one is tissue subclassification, and the third one is diagnosis. We have WHO grading of grade 1, grade 2, grade 3, etc. We can do that, and we have segmentation and prognosis. So these are the typical diagnosis tasks that can be accomplished in computational pathology. And the work that we are doing is about detection. So there's so many areas that there is work that is going to happen, and we are focusing only on the detection part. If you look at the background on computational histopathology, we have the research articles by organs. If you see there about, there's a research survey paper which surveyed 400 papers. Out of that, by brain, if you see, there are hardly 7 to 8 papers going by the classification, and breast cancers are very widely researched. This also shows an opportunity and a challenge. Histopathology in brain is at a, I would say, an embryonic stage where there's a lot of scope for research and ability for us to bring in good patient outcomes. So this is a data science flow for histopathology. So I don't know if it's visible. Yeah. So this is the data science flow. First, initially, the problem statement is done, and then there is scanning. I will talk about whole slide image scanning, where the slides are scanned, and then it is annotated by expert pathologists, the malignant areas, and the subtyping is done, and then we build AI models around that. After that, evaluation, this is the most critical part. If it's about 90% or 95% accuracy, doesn't mean anything until it is evaluated by expert pathologists. After this, we want to build models. This is a deployment, basically building products out of it. So we have, in this workflow, we have done work, basically one thread in this whole workflow. We have done work on detection of necrosis in meningioma, a type of malignancy of the human brain. So we are trying to get a thread out of this whole process. So we have done scanning and tile and region level annotation of necrosis by expert pathologists. After this, we have created a curated data set. Data sets, open source data sets do not mean anything until it is validated by experts. That's what we mean by curated data sets. That is where all the interdisciplinary work happens. The data set that we have created is validated by expert neuropathologists from NIMHANS, and that is the value addition that we are creating in terms of having a interdisciplinary research. After this, we have worked on building triple ITB from the data science group. We have built models for detection of meningioma using ML and deep nuclear net. We will talk about it. And after this, it is evaluated by the NIMHANS team, Dr. Anita and Dr. Shilpa Rao, whom we work closely with. They evaluate this. So we have taken a complete thread out of this pipeline, and I'll show the results out of that. So whole slide imaging, when compared to radio imaging, the primary source is images itself. In the case of pathology, we have slides, and there are scanners which have come up now. You will be able to scan the whole slide and create images out of it. Basically, we are bringing slides onto the digital world, which will enable us to do image processing and apply all our AI ML techniques. There are issues with this, in a sense, challenges. So whole slide images are digital slides when created when tissue slides are scanned by a microscope. The issue is it's not just one image, it has to be acquired at different scales, which means that these images are gigapixel images. Each image is going to be around 2 to 2.5 gigapixel, and we know that in deep learning we need several hundreds and thousands of samples. Just imagine 2 gigapixel into 1000 samples. We do not have enough computing power yet, so that poses a big challenge for us. In order to address that challenge, what we do is basically break the beast into monster into pieces and conquer it. So these images are acquired at different scales, and each image is broken into a tile or a patch, what we call a tile or a patch, typically 256 cross 256 pixels. So we break up, there are 2 dimensions to it. One is the scale at which it is acquired, and the other one, the number of tiles. Basically, if you see, you have a stack of tiles at this level of scale. Basically, this is 8x, 10x, and 40x magnification. At this level, we have this many slides, and as the magnification increases, number of tiles increases. So this is a constraint that we have to work with. We will have to work with tiles. We work with tiles, and this is an example of a whole slide image at magnification. So this is at 1 magnification. As you can see, if we keep on going at different level of scales, we can get to the level of cells here. This is at a very high level. As we go down deeper and deeper, this is an example where we go to the level of cells here. And this is an example of an annotation bar marked by an expert pathologist. This is a necrotic region. In those images, expert pathologists will mark the necrotic regions. So these are ground truths or a reference for us in building AI models. This is what we are going to learn. So this is an example of annotation by expert pathologists. If you compare the challenges of pathology here, computational pathology, it is easy to mark. If you do face detection and vehicle detection, anybody can do it. The challenge that this brings in here is it has to be done by expert pathologists here. And these are typical ATPR features, fascicle voles, necrosis, different type. So we focus only on one part of it. We focus only on the necrosis part. And as you understand, clinically, necrosis is relatively easier to detect. But they gave us, as part of machine learning, they said try to do this first. We'll look at other features later on. So we first started with necrosis. So we have some characteristics of WSI image processing. It's not the characteristic typical image processing that happens. 1 centimeter size of a tissue, biopsy tissue can produce a 4.8 GB image, which is 50,000 cross 50,000 pixels. And slides are scanned at various magnification. As I said, it can be scanned at different level of magnification based on what is to be detected. For example, if there are regions, its magnification is taken at 8 or 20x, for example, necrosis region. But if it is at a cell level, when you say, for example, if it is mitosis detection, it will be scaled at the region of 40x. So, and since we cannot load all of those images in one go, we'll do patch and tile level processing. Another issue that we have is vendor specific formats. There are multiple providers of the scanners. Each one have got their own extension, their .scn files, and even the type of staining. If you have HND staining, the HND stain, the same slide when scanned in different scanners will have different backgrounds, and then their file extensions are also different. Basically, I cannot load. There's one .scn file and .png files. It's not common across. And then, as I said, domain adaptation for slides. Each one will have, each scanner will have a different background. These are challenges that we have. And one good thing is there are open tools that are coming up, some tools like UUPath and OpenSlide, where it helps us load images from multiple vendors. And the data sets, we have, there are a few data sets in TCGA, even, I don't know, it's a very popular one. The TCGA has, it's a cancer archive where images and patient level, case level information is available for different organs. We use that extensively. So this is an example of patch generation. Now that we get a whole slide image which is scanned, this is a bigger one. But yeah, so what we have to do is when we generate tiles, we have to ensure that these artifacts are not covered. It's not that we just take a whole slide image and just generate the patch. We use image geometry to generate the patches. We start at offset 0, 0, and then keep on shifting by 256 or 256 non-intersecting patches. In the case of generating patches, we have to ensure that these artifacts, the empty spaces are not included. If we include those, they will skew the data sets. So as part of the tile generation itself, we will ensure that the empty spaces are removed and the tiles are generated. In this case, we have a curated meningioma data set. The good part is that this is the only data set is available for meningioma at this level, and the bad part is that this is the only data set available. So it means that there is lot of scope. There's a paucity of data. There is so much of scope for us to build data sets here and work on this domain. So we worked on 9 samples, and then we also have a label type. There are 2 types of annotation that is done. The region is given. Out of those region, we generate the tiles. The other part is we generate the tiles for the whole image, and we present it to the pathologist, and they say whether it is necrosis or not. So basically, annotation after tile and extracting tiles based on the region annotation. So we have, this is the data set that we have created. It's a curated one, expert validated. And there are clinical requirements of ML predictions. This is one point that we found from the interactions with the clinicians. It's not about accuracy at all. If you have a slide, if it is necrotic anywhere, it doesn't matter. It doesn't mean that I have to cover 95% of the necrotic area in a slide. It is about how small necrotic area that you can pick up. Is there something which a pathologist can because they keep staring at slides so many times. Is there a small set of piece of area of necrotic? You can determine, that is what is important. So based on that, we worked on metrics for that. In the sense, it is a needle and a haystack class imbalance problem. If you can, your objective is to detect the needle, not say that how much of hay is present, you detect it. So it's a needle in the haystack problem. So we define metrics saying that how many regions are there in a whole slide image, and within that region, can you pick up at least one tile? So which means it's sort of seeding for them. They can go zoom into it and work on it. Is there something that human error which can happen due to human error, can that be detected using this model? And it's not about accuracy only. We define metrics based on what is the clinical requirement. So we are going to present 2 works here. One is necrosis detection in meningioma using ML. This is done by one of our MS by research student. Here, as we know, machine learning, the difference between machine learning and deep learning is machine learning is handcrafted features. As a professor from IIT Mumbai very nicely said, when you look at it, whether you're looking at the eye or whether you're looking at the tree, so that work has to be done by us in machine learning, so which means we call handcrafted features. So in machine learning, we have these are the feature, we do the tile generation, we have positive and negative labels, malignant, necrotic or non-necrotic, and we do feature extraction. This feature extraction is again based on what, so it can be color histograms, it can be whatever, the GLCM metrics, the texture metrics, it can be the histograms and other things. So after this, we use ML classification algorithms, and we, as we said, we tried to, as usual, we tried a variety of algorithms, and we picked the best ones. And this is a visualization of prediction using machine learning algorithms. So this is a necrotic area, and these are the true positives, and true negatives are the benign area. So there are false positives and false negatives. One issue that we observed is, so if it doesn't perform well on the border tiles. If you say border tiles, which means it's a partially necrotic and partially non-necrotic. There are not enough features for it to perform well. So we found that all our false positives and false negatives was happening more on the border tiles. This is the issue we try to address in the subsequent work. And this is the recall performance on detection. If you see, I have not plotted accuracy at all. Accuracy we do, but the primary metric is recall. Recall, as we know, is a measure of false negatives. How good are you? It's TP by TP plus Fn. Fn is in denominator. How good are you in terms of not missing out a necrotic region? So we have, and then we have also tried to find area, percentage area, since the metric was to say how small an area you predict. So we found, we tried to categorize the tiles based on the percentage area in each of the tiles, and we have seen that if you see even the area of necrotic area within the tiles is less, the recall is less, and as the area increases, the recall increases. So here we can say that, you can say a threshold, you say the 50 to 60%, we can say if there is at least 50% necrotic area, we can say that our confidence of predicting a necrotic region is high. And if you see, we are trying to bring out the metrics based on the clinical requirements. So next one is, as we saw, we try to move to deep learning here for detection of meningioma, and we also moved to glioblastoma here. The key difference, as we saw here, we try to extract different features, and then we see iteratively very cumbersome work and find different features, different algorithms. So we try to do end-to-end learning here. So this is the method that we have used. One is, we call it deep nuclei net. This again work by an MS by research student. So we have tried to do domain adaptation. So this is an end-to-end learning. This is the standard exception net. What we did was, if you see, necrotic region is a function of the number of cells, number of dead cells. We found that for domain adaptation, what we did, we created one more model here, where we created, there is a technique called StarDist. We extracted the nuclei only. It's just like this, there were some issues with this, we'll talk about it. Based on this, we moved from ML to this deep learning method, and we found that the areas were still not good. So we tried to extract the nuclei and augment that. Basically, we tried to have a combination of these two models, and of course, we gave it to a SVM classifier later. And if you see the separation, this is T-SNEEZY feature visualization. If you see the separation with the other normal one, and this is the one which is proposed by our model, deep nuclei net, we see a clear separation which is happening. And the domain knowledge extraction, nuclei detection, as we saw, we tried to, the classical ones are semantic segmentation and object detection, as we all know, these do not work. We tried with this, these do not work. And there is another technique called nuclei segmentation using StarDist, where the other issue with this here is, in case of, in this case, when you try to do object detection, it will not be able to detect overlying objects. In order to address that part, we use what we call StarDist nuclei segmentation. It finds the probability of the centroid of that object, and then you have a sort of star convex distances from the nuclei center of the part. You join all these edges, you will be able to get that object. With this new technique, we were able to adapt, and then we were able to differentiate overlapping cells as well. So this is detection performance on boundary tiles. Again, as we said, we saw that it was not performing well on boundary tiles. We tried to categorize the data set based on boundary, and we found that one is with completely within the tile, completely within all 100% necrosis and partial necrosis. We have seen that. We have seen that when the recall is greater than 50%, when the region increases, that correlates with us. And another thing that we did was, we trained on meningioma, and we used GBM Dr. Rose was speaking about glioblastoma on the radionomic side. We are trying to do it on the histopathology side. The TCGA has glioblastoma slides. We trained on meningioma and just try to predict. We want to know if the same models can be used across different brain tissues. We found that accuracy was fairly good in terms of GBM. Again, this was validated. This is an unseen sample prediction that we did. It was validated by doctors from NIMHANS, and this is the results that we have. And I will talk about, I have 5 minutes, talk about explainability and bias. So we spoke a lot about explainability and bias. We have developed some technique, we have used some techniques to identify quickly. If you see, there is a model which detects doctors and nurses. The biased model is looking at just faces and trying to predict whether it is a doctor or nurse, because it generated bias saying that all males are doctors and all females are nurses, while actually it should have looked at the stet and the coat. We expected the model to be looking at the person is having a stet or a half sleeve, but predictions are right, accuracy is good. But this is what we call is all about explainability. There is a technique called GradCam visualization. This will help us identify if there is any bias or basically bias in the network. So what we did was we applied that here. So here explainability and bias, the same concept we used here. So this is a necrotic prediction. So this is something which clinicians will be able to explain. We will see that it is predicted necrosis based on these regions. Is it right? This will help us have an interesting conversations with the clinicians. And we said this is a non-necrotic region. And if we see that the prominent cells are picked up here, is it calling it non-necrosis because there are cells here? Is it calling it necrosis because there are no cells here, but these regions? This is something that we will not be able to answer, something the clinicians will be able to answer. So this is the type of conversation we'll have in terms of explainability and bias. We adapted those GradCam techniques here for our work. So in future work is we have just 8 or 10 slides. We're going to enhance this for brain tissue types and other brain tissues and other subtyping also. So creating a tool to realize the WISI processing, so because we want to help NIMHANS have something which will be of use to them. We are trying to build a pipeline which they can use. And of course, model development, we'll have to do tissue classification, grading and prognosis. Emerging trends in computational pathology is very interesting. We know that large language models are with us. Professor spoke about zero-shot learning, CLIP. Basically, instead of having large data sets, you have a set of samples which you learn. Basically, it's a few-shot learning or zero-shot learning that can be done. And another important development that's happening is instead of having handcrafted, basically marking the annotation, given that pathologists' time are very critical, can we look at medical records? Now that we have LLMs, we can map text to images. Can we look at the descriptions in the diagnostic reports and we use that to create images to do the annotation is another thing. Of course, in visual transformers, we have patch individually, but that is not how a doctor sees. They look at the region. So there is no spatial relationship between each of the patches. Each one of them are looked independently. So there are techniques for that. Vision transformers, if you see, it does positional encoding. We'll try to see if we can use those techniques. And of course, multimodal large language models, it's opening up a lot of opportunities for us. It's interesting. So yeah, so these are the references. That's all I had to say. Any questions I can take? So we look at a lot of radionomy data. So we have taken a path to go into the pathology side of it. Hopefully it will create interest. It's an area to be explored. You can reach us at IIITV. You can reach out to us if there's any collaborative work that needs to be done. Thank you.
Long live India and France friendship.